Hey, it's Matt Roush.
And Mike Brennan.
And we're back with another segment of the M Squared TechCast. You know him, you love him. He's been our guest since way back in the spring when all this madness first started. It is our very own epidemiologist and infectious disease expert, Fred Brown. Fred, this is kind of a red letter day that we have you here on. This is the first day in the United States that the Pfizer BioNTech vaccine is being administered to people outside of a study.
Yes.
Yes. So I guess this is good news, but there's this is the first crack of dawn in the astronomical twilight here, right? It's still pretty dark out there.
I like that. That's a good metaphor.
It's dark, and before the light always is. It's a little bit darker.
Yes.
So we're going in a bad direction. But the good news is this is probably one of the best vaccine study results I've ever seen. And I've been in this business a long time.
Yeah.
I thought I'd go. If you like, I can go through that with you. I have a little presentation I put together based on everything that was presented to me and the FDA over the course of the last two weeks, and I have a little collection of slides. Would you like to take a look and see what we have?
Sure.
Look at or
yeah, absolutely.
While you're spooling that up,
yes.
Over the weekend, everyone was bragging about how quickly that this vaccine was developed. But then they were saying part of it was they had the sort of the basic framework from the Sars vaccine that was developed 18 years ago. And this falls into the same family. And this is not my area of expertise, it's yours. Are they correct?
They're right. Yeah, this is based on a lot of research that we've been able to do over the last 20 years. And SARS is also a coronavirus, very closely related. This is Sars Coronavirus 2. So SARS 1 is as close as about 85% consistent with SARS 2. So if you look at the gene code, and so it's very similar, and we were able to use the same vector technologies, the same mRNA technologies for SARS 1 as we did for SARS 2. The sad thing about those vaccines we developed for SARS 1 is that nothing we've developed so far is durable for more than 18 months. So that's something we really have to watch for in the future. But these results are fabulous, and I thought I don't know if you guys can see my screen okay.
Yeah.
Yep.
I'm using PDF files this time, so it's not going to be as smooth possibly as what I'm in PowerPoint. But this was what was put in front of VRBPAC. So VRBPAC is the group that actually advises. It's the advisory council to the Vaccines and Biologicals Council that advises the FDA about whether we should go forward or not. And at the end of the day, I believe we've got 21 people on it, something like that, on this particular one to kind of look at everything. These are mostly professors. They certainly have to announce any conflicts of interest they have before they go on. Some of them own a few shares of Pfizer stock. A few of them had made some had advised other companies in the course of their careers, but no one has any conflicts of interest. Everyone looks completely objectively. Most of them are medical doctors, PhDs at universities. And so they're looking at this stuff very closely, and this was a question that was put to the vote. This is on the 10th of December. So the question, yes or no, is basically a risk-benefit analysis based on what these experts think about the data they've seen. And so the question they had before them was, based on the totality of all the scientific evidence available, do the benefits of Pfizer BioNTech COVID-19 vaccine outweigh its risks for use in individuals 16 years of age and older? So this is what was presented. Basically, here's the whole process that Pfizer went through. They went through, and this is, as you said, very rapid. And the reason it was rapid is, number one, our research program was largely developed. We only had a couple of days to create something that was suitable for SARS-CoV-2 or the COVID based on SARS. That was Moderna. Pfizer took a little longer. They ended up, Moderna had something ready to go after 2 days. Pfizer took, well, you can see a letter of intent with BioNTech, and then they had to choose based on phase 1 results which candidate they wanted to take. And they had 4 candidates, and they took on July 24th. So this is January, January, as I recall, 10th when the genetic code was announced. They took January, February, March, April, May, June, and July. So it's almost 6 months to figure out slightly over 6 months to decide which of 4 candidates you're going to take. And let's take a look now. And what we do is we go through the genetic sequence that was announced as this arrow is a little bit overly pessimistic. It was actually announced, I believe, on January 10th. We did the animal studies literally within a month or two, which is pretty quick, but don't forget we were looking at existing technologies and confirming results. And then we had the process development, which the United States paid for in most cases, not in the case of Pfizer, but in most cases we did that. And we went right into phase 1 in April. And then we went into pivotal phase 2-3 trials based on the results of those phase 1 trials in July. So let's take a look at phase 1 trial results. And here, what they were trying to do is they were trying to show that the people who took their vaccine had 2 kinds of immune response. The first kind of immune response is antibody circulation. So did you have neutralizing antibodies in your blood after you took the vaccine? And here you can see what happened. This was, and they did this for four different kinds of drug. They chose this one, BNT 162b2, and you can see between the ages of 18 and 55, we see that they had different categories and different days. Here's day 52. Here, so this is after about a month here, and you can start to see after dosing. So nothing here day 1. You'd expect that. You had day 21. You had some titer, and then at day 28, you had quite a bit of titer, and day 35 and 40, you can start to see it. So peaks up and then comes back down again, which is what you expect. HCS is how much is in the population generally, and so you can see that basically these are for the when you're infected. So what happened is we were able to demonstrate definitively that we had as much antibody titer in our bodies after taking the vaccine as people who were infected did. You see that? So that's what that shows. And it didn't matter whether you were 18 or 85, you still had an immune response. A little bit lower in the 85, 65 to 85, but still really good and very consistent with what we see in nature. That's the first question. Do you have antibodies kind of moving around your body that are going to attack the virus before it can attack you? And that's always a good thing to have, because you want to block the virus as much as you can. So the great news is we have those floating antibodies, but we don't think that's enough. Typically in coronavirus, you need to have not just the floating antibodies, you also have to have memory antibodies. So your body, when these memory antibodies detect COVID coming in, they actually will produce much more antibody. And so that's what they call CD4 and CD8 levels. And so we also looked at those. And here you see the same thing. Not only is when you get, we're having a 30 microgram dose here, you can see here's what happens naturally. And here on the CD4 cells, those are memory T cells, you get not only, it's actually a better response than if you were infected. This is not normal. Most times with vaccines, you get about the same or slightly less. But in this case, our memory was better on the vaccine than it was even when you had the real disease. And the same thing's true about CD8. These are priming for killer T cells, and you can see same kinds of things, kind of a 10-fold increase over natural what's naturally there in some cases. So that was tremendous. And so we were expecting some really great results. And you can see we even started to look at 4 different kinds of antibodies. This is the interleukin family that we were looking at to see which of these antibodies are most neutralizing. So that's what happened at the end of phase 1. And so based on that, we said, hey, we should be able now to go into human trials. We've done the animal studies. Everything worked out well. We did mice, we did non-human primates. All of them showed good efficacy, even with challenge tests, which means you give them the vaccine and then you breathe the bad virus onto them, and even then people were these non-human primates were protected. We went into humans, we did that. We showed the immune response was great at a statistically significant level. And so in June, July, they said, we want to test, initially they said 30,000 people. And they enrolled the 30,000 people through September, and they said, you know what, it's not, we'd like to do more with the elderly and more with minority patients, and so we're going to supercharge our study with the elderly and minority patients to really make sure we've got something that's good across all populations and all demographies. And so they went to 44,000 healthy subjects, and there were some inclusion exclusion criteria which we'll go through, and you should take those to heart. And we wanted a balanced racial ethnicity profile, which you'll see in a second. We did exclude immunocompromised. We did exclude pregnant females. We did exclude those under 16. We did exclude those people who had adverse events, adverse allergies, people who were taking EpiPen with them when they take their shots because they're known to have bad reactions. So those were some of the excluded people. But you can see here, the demography was really strong. For 44,000 people, 21,000 people got the actual drug, and 21,000 people got the placebo. And you can see basically it's about a 50-50 split for male-female. These are randomized. So what happens is the doctor will say, you should inject patient number 6 with this syringe, and the doctor will take that syringe without knowing what is in that syringe or who the person is who he's treating. That is completely randomized. They'll inject that person, and then we'll find out what happens. And sometimes the person will have gotten placebo, just the saline solution. Other times they'll have the active ingredient. And then we look at the differences. That's called a double-blinded, placebo-controlled clinical trial. That's the way we designed this test, and those are by far the most sensitive and specific trials that we've got to use to decide whether something's efficacious or not. So here's the breakdown. What do you guys think? You happy with the breakdown?
Yeah.
You think it's pretty fair?
Mm-hmm.
Well, not being an expert in these sorts of things. It's a little bit below the societal representation of Black folks altogether, which I think is like 13-14%, but It's 10%, so that's reasonably close, I guess. Yeah,
almost 4,000, as I recall, in the 4,000 range. A little over 4,000, excuse me, for 4,200 Black or African American people were in the trial. And that's 4,000 people is a lot of people. We frequently will do a vaccine trial with just 2,000 or 3,000, depending on how prevalent it is and what the infection rates are and so on in the area where we're doing the injection. So 4,000 people is statistically significant, and you can even break it down to that. You can even start to look at people of an ethnicity plus their health risks and so on. And so when we do that, you can see that Pfizer's really happy about the fact they got a lot of elderly in this trial, right? Because we were really worried they're the people who are most susceptible. And if we'd had serious adverse events that overwhelmed the benefit of the vaccine in this age group, we would have had a big problem. But you can see basically they're highly represented, and you'll see in a second the results. That's the demography. So just remember that when you watch the rest of the slides, if you can, at a high level. The one area where we are weak, frankly, was the age 16 to 18. We only had about 100 people in that age group. And so you saw those four people who voted against allowing the 16-year-olds to be vaccinated. They didn't think that was a good idea out of the total committee. That was because we were underrepresented in that age group. We just didn't have that many of them. Here's how the efficacy analysis worked. You can see that they had you got your drug 21 days, and then you got your drug, and 21 days later you got your second dose, and then they're going to follow these people for 2 years. That includes safety as well. They're going to look at safety after 2 months and after 6 months. After the 6 months are over, they will then apply for what they call a BLA. This is for an EUA. Emergency use authorization. The BLA is what most drugs have. That's a biological license application. That actually then permits that's the full licensure. The emergency one is just for cases like this where we have.
Well, I do like the little diagrams on the heads there on what they're being followed for. It looks like the one on the left is severely thinning hair on the top of the head.
Well, you
I'm
know,
not quite sure what the other ones are.
we do this so there's no so the people understand. You know, there were a lot of people who are speaking lots of different languages in the test, and so it's important we've got
the one on the left
symbols.
looks like a bad comb over, but go on. So
and so when we do our consent part of that is making sure people know what they're getting themselves into, and so everyone had to sign a consent saying, yes, I agree that I want to be part of this trial, and that was part of the trial. We had, you know, show pictures sometimes to some people just to make sure they really understood, and that's true about young children as well. So then we had the primary endpoints. And so here you can see that the endpoint case was if you had one of these symptoms, right? And it could be very mild. You could have a mild fever or a mild cough or a little bit of chill. You say, no, I don't feel that great. I'm going to go to the doctor. And if you then tested positive in a central laboratory, PCR, which is our most sensitive test, and the test said, yep, you've got COVID, at that point, you'd be set aside and say, okay, we're going to monitor this person because we're not sure whether that person has placebo or whether it has active injected, right? So we're going to take a look, and when we break open the code, we'll know whether that person was injected with the real drug or whether it was just a saline solution. So that's what we do. And you'll see that there are a lot of people who thought they got sick, but actually weren't. Based on the PCR test. So based on PCR test confirmation, this is largely, you know, kind of you feeling a little bit like you have the flu, plus a little bit more. So then here's what happened. So the efficacy, we found 8. So we broke open that big collection. There were 170 people who felt sick and then were confirmed. Of that 170, we found 8 that actually had been injected and 162 who had the placebo. Now, if our test wasn't working, you'd expect that to be 50-50, right? Each group having 85 in that case out of 170. But we found only 8 with the vaccine, which means that the VE, which is the vaccine efficacy, you know, in this case was 95%. And we're very, very confident. The confidence interval for 95% confidence is 90 to 97%. So we're in that range of true standard deviations of being either at 90% or 97%, 98% of all of this happening by. There's almost a no chance of this happening by chance, right? It's the p-values are fabulous. So we think this is an extremely effective vaccine. Even if you took the very worst possible scenarios of the analysis, it's still an incredibly effective vaccine. The best vaccines we have in the world today are measles vaccines. They're 98%. Everything I've worked on has been less than 90%, and these are for childhood diseases and so on. So this is an extremely good outcome. And here is the breakdown. So if you're worried about, whether those infections, whether it was less effective in, for example, people who are less than 64 years old or over 65, you can see that in fact the efficacy down here still always is above 90 or so percent, with the exception of all others races. And some of the results that we had in Brazil. But you can see that we're talking about something that's incredibly effective at a very, very high confidence level based on the results. And part of the results of Brazil was we couldn't, if we'd had more people, this is 1 in 8, 1 in 9, we would have shown the efficacy, I think. What you really want to look at is these high numbers where you've got 7 out of 153 or 7 out of 150. Those are much larger numbers, and so we're much more confident about those numbers. So we think it's in the 95% range.
I noticed nobody under 18 were anybody under 18 tested? I can't quite tell here.
Yes, there were about 150 people, as I recall. Slightly over 100 people were between 16 and 18.
Okay.
Yeah.
Now, is it not something that you want to give anybody younger than 16, or just you're not sure yet?
We don't know yet. We haven't done the tests, right?
Okay.
So as soon as we do the tests, and that's being planned now, and hopefully we'll have those test results by the end of the summer. It depends. It depends on whether we're able to kind of create what they call bridging studies or not. The bridging studies show quite a bit of difference between a childhood immune response and an adult immune response. We'll actually have to reconduct a full set of clinical trials. We can't apply what we've learned in these clinical trials of the over 18s to those who are under 18 because their immune systems are responding differently to the vaccine. We don't think that's going to be the case and that we will be able to use bridging studies, but the worst possible scenario is, we'd have to do, what they call real world trials based on observation on the children. And that'll take probably, July, August timeframe, at which point, hopefully they'll be ready for school.
That's what I was driving at is the grade school kids. Even my grandson, although he's only 2, but my daughter wants to put him in preschool in another year or 2. And I remember from my days when my daughter was in preschool, my wife and I were constantly sick. So.
Yep. Yeah. It's that's the petri dish. I've got a whole, Mike, on my website, one of my friends wanted to put their kids into preschool and wanted to know what screening criteria there were, and I'll send it over to you, just to hear the screening criteria for children. It's important they have well, anyway, I'll send it over. We can go. Why don't we go through it next week? How does that sound?
Sure, it sounds great.
Yeah, we'll talk a little bit about that.
Because I think a lot of the parents are getting really tired of being the teacher, and they'd like to see their kids go back to school, right?
These kids have a lot of energy, don't they?
Yeah. So
we were interested also in efficacy with regard to whether you were sick, and so here in this case, in the general population, 41% of us have some kind of comorbidity. So you can see that about half of the people who were on and about 41%, sure enough, were who had the placebo, had no comorbidity versus some having some comorbidity. You can see that even if you had a bad comorbidity, and again, the number of cancer, we just didn't have enough at all in the overall sample. I think if we had more cancer survivors, we would have had a higher number here. But we didn't have that many in. But other than that one group, you can see that basically, even if you've got a bad comorbidity, you're still at 95% efficacy, which is great news, obviously, because the people with comorbidity, which we'll get into in a little bit, are at a much higher. If you've got 2 or 3 of these comorbidities, you're at a 7 times higher chance of dying if you get the disease. So it's great that these people are having good outcomes being on the vaccine. Here we've got the overall numbers with or without evidence of infection prior to 7 days. You can see that this is a secondary endpoint. So the primary endpoints were only those without evidence of infection. We actually did test for people who did have some evidence of infection, and you can see. It's important that even if you've had COVID, get this vaccine because it's that much more effective for you in this. If you've had the disease. So here are the case definitions we went through. You can see that here is what a normal kind of what they call a mild or a disease outcome is. And then we also looked at severe outcomes. And this is probably the most important because if you've got severe outcomes, including respiratory failure, shock, severe systemic illnesses, or you're on your way to dying through ICU admission, you want to be able to slow that down. And what we found was, that we do protect against severe disease. Now, the numbers aren't that big, but they're incredibly significant because don't forget, we tested 44,000 people. So you can get to a number that looks like this. Out of 17,000 people, one of them got who had the actual drug. One of them got what they call severe COVID. This one person was asked about quite a bit because we were concerned about what kind of person this person was. And it turns out that her severe adverse event did not cause hospitalization. She was she had a pulse oximeter, and her pulse oximeter read 93 at one point. And 93 pulse oximetry, frankly, my pulse oximetry runs at around 93. So it's a so she did go to 93, and for that reason, she was cited to have a severe adverse event. Now, the people on placebo, their experience was a little bit different. I think nine of them, they say 14, I say. So efficacy and first severe, 14 people. Of these 14, 9 of them were hospitalized. Of those 9, 6 of them went into the ICU, and 3 of them were put on ventilators. So you can, so you want to be on this vaccine. You do not want to be one of those people on the ventilators, right? And so it's a very, very significant finding that we are finding efficacy even against the most severe cases of COVID. And here is the money shot, right? The money shot says that if you look at the cumulative instances of COVID, which population would you rather be in? Would you rather be in those that have, a 2% cumulative occurrence? Or would you rather be in one that has a 0.002% occurrence, right? And you'd rather be in this group. And in fact, when you look even closer, what we find is that despite those titer levels that you saw that showed the importance of having a second dose, even after, I'm sorry, let me go down one, apologies. If you blow up, I'm sorry, I jumped around a little bit here. If you blow up that little area down between 7 and 21 days, look at that separation, right? We're just climbing up at about the same rate as whether you've had the shot or not. Day 9, it's absolutely parallel, right? Day 10, parallel. Day 11, parallel. Day 12, complete separation occurs. It never comes back. After about 12 days, you're in pretty good shape to be what we think, to avoid COVID completely. So that's that result, which I thought, was incredibly significant. The reason that it may be significant is we may be able to get to 1 dose. Right now, that's not recommended. We are going to hit everybody with 2 doses because you saw of the result of phase 1 trials, which showed that number of antibodies increasing so much with a second dose. And that the reason that's important is because we might be protected, but we want to be protected for a long time. Like I said, our longest experience is 1.5 years of duration for these kinds of vaccines. We'd love it if we get way up there on the immuno curve, and then have a lot longer time to come back down and be protected for that much longer, so you avoid the next booster shot. So that's the idea of having that second booster, and why it's going to be so important to have that second booster, we think.
Now, this is the Pfizer trials. Have you done anything with Moderna yet?
I do. I have, and that will all come out this week. Actually, on Wednesday, it'll all be published.
All right.
I have a little bit of information beyond that. Moderna, at a high level, this isn't conclusive yet because it's not officially presented, but the unofficial numbers indicate that Moderna has fewer side effects, and we'll get into that in a second. And it's of course easier to transport. It's -20, not -70. And there's some positive things about the Moderna drug that I think it might make it a little bit better than the Pfizer drug even.
Well, and more readily available. I saw one broadcast that said that Pfizer has only 30 million doses for the U.S. or some number like that, and they're under contract. Then to the next batch has to go to Europe or other places like that, correct. Right?
Yeah, that's right. A lot of these are split, right? So Sanofi's, we get a lot of AstraZeneca drug, 300 million doses, but 600 million are going over to Europe and other parts of the world. So you've got to watch the splitting. And of course, it's a global supply chain, so that actually helps. Believe it or not, it's better to vaccinate the world than it is just one country, because that one country then is, depending on the kind of vaccine you've got, can really get a bad bounce back effect if everyone around them is coming down with COVID all over the place. Then the COVID will come back to your country too if you haven't, unless you really impact transmission, and we haven't been able to demonstrate that yet. So this is, let me go down a little bit further and talk about some safety. So here's how we do safety. So we talked about efficacy, now we're talking about safety. And you can see we talk about 2 different things. One is a reactogenicity subset. What this means is it means that whenever you have a shot of any kind that's effective, you're going to have some reaction from your immune system. So actually, reactive immunogenicity means you don't feel so good, but it may not be a bad thing that you're having some kind of reaction, because it means it's actually working and stimulating your immune system. Then we had another group we looked at 2 month post dose. Remember, that was the new requirement by the FDA. They originally said just 1 month's enough for us, thank you very much. And then they said, nah, we better take 2. So total number of patients were 43,000 patients, but only 37,000 of them had the 2 month window of observation. And we have to go through and continue on and find and continue to observe the other 6,000 that are remaining. But still, you'll see that we've split the group basically into 2 areas, one of 8,000 subjects and the other with 37,000 subjects, of whom 19,000 actually had our drug. So we're looking at, when you look at these adverse events, you're looking at the results from 19,000 people generally, or 8,000 people who are actually asked at a regular period, how are you feeling today? How are you feeling today? So let's take a look on that. And the answer was, Doc, I feel pretty good after day 1, but at day 2, we don't feel very good. You can see that day 2, they started to feel a lot of, a little bit of fever. A lot of them had fatigue, right? About half of them had fatigue. Some had headaches, some had chills. And don't forget, this is, and you can see basically the over 55-year-olds actually had, their immune systems aren't quite as good, so they had less of this effect than the younger groups. But still, you're going to feel tired. You might have a little headache. Usually take some Tylenol and it went away. You might have some chills. A little bit of muscle pain, and that changed on dose 1 versus dose 2. So you can take a look at, I'm sorry, here's the placebo numbers. And you can see what's interesting is people had these effects anyway, whether they were on the drug or they weren't on the drug, right? So it turns out that you've got to reduce these numbers a little bit, like fatigue. Well, right? So the 50%, 60% of people said that we got some fatigue. But of those, there was at least a couple of people who had extreme fatigue or so-called severe fatigue in the placebo group. So if you start to, weight average the results, you'll see that actually in this case, even just going through the experiment gives you a little bit of fatigue and headache and chills. And so correcting for that, the big thing is this chill area. You can see that is significant, and muscle pain. Those are significantly different than the placebo group. With headache and fatigue, we'll have a little bit of it, but the things that will really be different are the chills and the muscle pain. And then we will.
Well, that's, I mean, a lot of shots. I've recently had a tetanus shot, and the 2-shot, the 2-shot. Oh shoot, I'm blanking on the name of the ailment regimen, and both of those, my arm was sore as heck for days after.
Yeah.
That was the 2-shot shingles thing.
The shingle shot will get you.
I felt like crap after the second one. I really did.
Oh well, the second one is going to be a little bit of a worse effect, as you saw from the numbers, than the first one.
Yeah.
Here, what we're really interested in, ultimately, you're going to have a lot of people who have sore arms.
Mm-hmm.
So that was the most reported piece. And then there was, but what we're really interested in is, does it have any systemic effects on your organ systems? And you can see that based on people who got the drug and didn't get the drug, there's almost no impact, right? The placebo group, this is why it's so important to have a placebo group, right? Because you don't know whether just going through the experiment causes some of these effects or not. And in this case, you can see that cardiac disorders were the same, whether you got a saline solution or whether you got the active. Nervous system disorder was the same, whether you got basically a placebo or not. Neoplasms, again, the same, and the same thing about, turns out if you were, other complications were even worse than the placebo group and in the other group, in the active group. So this is the most important thing, and that basically we're seeing that this is a completely safe vaccine. It's really a remarkably safe vaccine There were lots of questions about whether this vaccine, if there were what they call reverse transcriptase complications, or that would mean that the RNA suddenly intercalated with your own DNA and caused problems. Nothing like that ever occurred. We were concerned about what they call ADE, which is antibody dependent enhancement, which means you get a small impact of the vaccine. It then selects for the worst possible virus in the world, and you get that virus, and that happens with dengue fever, for example. Our shots for dengue fever, and that's not a good effect. We're watching for that very carefully. That's why AstraZeneca was shut down for as long as it was. We were concerned about ADE in central nervous system infection, and in fact, we've now decided that there was none at all. That's a different drug, by the way. It has nothing to do with Pfizer's drug. Different technology, different everything. Here's the death. More people died getting saline injection than than on the drug. There were some issues. Bell's palsy. We had 4 cases of Bell's palsy in the active group, none in the placebo group, and this is an immune response. Again, 4 in 20,000 is something that is expected, and sometimes usually it spontaneously remits. But we are watching that now as well. So basically, highly tolerable, no significant safety findings at all in either group. So you can see we've hit all the primary objectives. We've hit all the secondary objectives. We hit 93% effectiveness rate. I mean, this is about as good as it gets, right? If you're taking a course in advanced calculus, you'd like to have this kind of a score, right? Per FDA definition, for CDC definition, for WHO definitions, the results were just overwhelming that we have a very, very good vaccine here, and 95% efficacy with no safety concerns. So that's as good as it gets. There is one group, of course, that is still, we are going to do some more work. We're going to look at hospitalization endpoints, ED endpoints, and then we're going to start to look at more specific populations. You mentioned earlier, Matt, about race and ethnicity. You wanted to have more. We'll do that. We'll do more nursing home residents work. We'll do more healthcare worker. We want to understand more about vaccine efficacy in the real world, because this is highly controlled. Everyone's saying, "Hey, sign me up. I'm interested." They go through selection processes. Usually, when we go into the real world, people will be slightly older, slightly sicker than the people who are accepted, have fewer allergies and so on. And so we'll look at that. Pfizer wants to look at, oops, I'm sorry about that, boostability. I'm going the wrong way, apologies. They want to look at boostability, to understand that better based on real levels in people, what their immune response has been. They want to look at dose ranging studies in pediatrics, worried about the children. Want to look at pregnancy. We want to look at the immunocompromised, people who are taking drugs because they're on cancer chemotherapies. They want to look at a second generation formulation like that Moderna has, that is more refrigerator stable. And they also want to look at a single big kind of capability for both flu and COVID at the same time. They're seeing every year. So that's what they're thinking about. And what I thought, now we only have a few minutes. One last population that's of interest, of course, is people who are still enrolled, right? What they decided to do was they decided to say, fine, we're going to issue the EUA, the emergency use authorization. If you're in a trial and you want to know whether you're getting a placebo or a real dose, you can do that. Or you can continue on the trial until a vaccine becomes available in your area, and then you get the vaccine. At that point, we will break open the code and let you know whether you've got a real one or not. And then if you don't have a real one, we will inject you and 21 days later put you on dose 4. That will force us to slowly lose the placebo group, but it's the ethical thing to do. I mean, if you've been happy to sign up, then it's the right thing to do, right? Just to give these guys the right thing at the top of as soon as they become eligible for it in their normal population. So that's that. And the result was that the FDA agreed that they had a good drug, vaccine, and they were going to approve the EUA, which they did, and now it is shipping. this was their conclusion.
Came out of Portage yesterday. I watched it Sunday morning. So.
Well, I didn't talk about some of the discussions about why we chose the groups that we did. I can do that next week.
We only got 4 minutes left. So
would you like to see it quickly? I can do that quickly. And why we selected.
If you could do a quickie.
I think I can do it pretty quick. So here we go. I will do that. And basically, the reason that we chose the people that we did were, if you look at seroprevalence, you'd think that a whole world had COVID, right? It turns out that if you look at what really is truly seroprevalence, in Michigan, only about less than 10% of us actually have antibodies to COVID. So if we just let this thing go, it's going to take us an awful long time without a vaccine to get healthy through natural immunity. You can see New York has the highest; they're at 23%, and in most states in the Midwest are at between are kind of 2 to 3%. Even though we've had this many deaths, even though we've had this, so we've got a long way to go if we want to kind of rely on natural immunity. So the question becomes, okay, fine, who gets the vaccine first? And the answer, this thing jumps on me. I'm sorry. The answer is, if you look at people who are hospitalized most, it's the people who are in the 65 plus. Even though most people who have the disease are between the ages of 18 and 49, the vast majority of those, you have a 4 times better chance of getting hospitalized if you're over 65. So that's a population that we want to vaccinate first, because the people who have who are walking around young don't have these symptoms.
Well, wouldn't you know it? I'm 64. So
not quite.
Sorry, Matt. You lose.
Yeah, you're in the be happy.
Back of the line.
Not much. Yeah.
It turns out the difference between being 60 and 65 and over 70 is like a 3-fold increase in death rate. So be happy you're in that lower age. Here's the hospitalization numbers. We really want to start to watch those 65 and olders. You can see they're less hospitalized now because they're staying away and trying to be more careful with their masking. But we still have an awful lot in that group. And so those are the people who are likely to overwhelm the hospitals by having too many people come. And if we overwhelm the hospitals, then we know the death rates are 2 to 14 times what they are if you don't overwhelm the hospitals. And so we also know that Hispanic, Black are much more susceptible and might go to the hospital a lot often. Again, we want to watch that overwhelming the hospital impact. So we're going to preferentially vaccinate the Hispanic, Latino, and the Black communities because that makes the most sense for our health system. Also makes more sense for them. If you're Black, 4 times higher chance of dying than if you're white. If you're Latino, 3 times higher chance of dying if you're Latino than if you're white.
I'm gonna have to give you the 1 minute warning, Fred.
1 minute warning. Here we go. You ready for the big? We'll go into. So here are your chances of dying if you're in your age group. 10-fold higher if you are if you're over 85 years if you get this disease than if you are an average age. If you're male, 30% higher chance of dying. If you're immunosuppressed, 40% higher chance of dying. These are big numbers on a population basis, but these are the ones, the people in the red box are the ones we want to get to first because they're going to be most impacted. We also want to watch out for their underlying medical conditions. If you're severely obese, 4 times higher chance of dying than if you don't. And so diabetes and kidney disease are the other big indicators. Coronary disease isn't good, but at least it isn't quite as high as these other disease types. And if you start adding them all together, huge impacts, right? 5 times higher. If you've got 3 of these comorbidity things collectively, you've got a big problem. That's why we're selecting people who are older and people who are in healthcare. And here, if you're a nurse in healthcare, that's 6% of the population. About 30% of the people who are in healthcare are nurses. Those are the people who are really most likely to catch the disease. And for that reason, we're starting with nurses and we're starting with elderly with comorbid conditions. So hopefully that makes sense.
Additional 4-year students love Lawrence Technological University's thriving campus life, but LTU has always met non-traditional students' needs too. Lawrence Tech offers over 100 degree and certificate programs that can get adult students started or back on track. And most of our classes are conveniently offered evenings at our beautiful Southfield campus or online, so you can balance your social, family, and work life even while you power up your career. Lawrence Tech, where Blue Devils dare.